introduce selection. The problem is, one, that doesn't apply to the majority of inversions out there. And two, even if it did, there are two additional mutational biases that you have to posit to actually get the serial inverted chromosomes that we see. And we have no evidence whatsoever for those mutational biases. So I have a simple alternative. The problem is the alternative is not very intuitive. So I'm going to come about it sideways. So first I'm going to introduce you to a cytogenetic pattern. Right? So here's the pattern. And once that's established, I'm going to tell you about some facts of Drosophila female meiosis. This might be boring to most of you guys, but it'll be very important. And then what I'm going to do is actually include population genetics. So that's why I'm in this session here. We're going to take a classic model, incorporate those cytogenetic mechanisms into the population genetic model, and we'll actually show that we can reproduce the pattern found in nature. So here's a chromosome, centromere on the left, telomere on the right. That's an inversion, just a rearrangement of the gene order. And this is a serially inverted chromosome. That's the second inversion on the same background that carried the first one. And from this, you can actually see two really important things. One, with just two mutations, you can actually get a very complex rearrangement. A rearrangement that has two inverted sections. Let's see if we can show those here, right here. Two different inverted sections, and a homosequential section that's been displaced down the chromosome. Now, if you were to just see the serial inverted chromosome out in nature, one of the great things about this is that you would actually know what steps had to be taken to create this. And that's one of the things that Sturman figured out. And when he did that, he was able to construct the first phylogeny based on genetic information. This is the gene arrangements of Drosophila pseudo-obscura, updated since Sturman's time, of course. Now this is most definitely an unrooted tree. This is all within species variation. But we can use information from frequency, arrangements in other species, geographic distributions to figure out what the ancestral arrangement is here in red, and actually polarize the tree. Now this is actually better than the average phylogenetic tree. It's actually a phylogenetic series because all of those dots are segregating in populations, and we have the exact, unambiguous ancestor-descendant relationship amongst all the relationships. Okay. So what we can actually do is call this a phylogenetic series, and what we'll do is we'll say, we'll rank something in a phylogenetic series. The ancestral has a ranking of 0. The first inversions are ranked 1, and the serial inversions ranked 2, 3, 4 on that way. Now, if you look at the characteristics of all those inversions that make up those arrangements along the chromosome here, not particularly striking. Maybe there's some clumping down near the distal end. But if you include some of the information about the age of the inversions, that is, Santa Cruz here is one of the earliest arrangements, and all of these are the arrangements that occurred subsequent on Santa Cruz's background. You can see that they're all kind of distally shifted. This is still a confusing graph. So, I should say, this is not the original observation. Nowitzki made this observation for about 7 arrangements a long time ago in one species, and everybody's kind of forgot about it since. So what I'm going to do is expand this to 6 more species of the obscura group, multiple chromosomes, multiple phylogenetic series, 134 arrangements. One of the problems is now that we have to standardize across chromosomes, so we're going to make everybody 100 unit lengths. The original observations are in cytogenetic bands. And on that scale, you can actually say something about inversions. You can say their size, their position, breakpoint locations, so on and so forth. There's a second standardization that we need to do, because as you create your series, you need to anchor it to the first inversion. So we're actually going to scale everything in a series back to the midpoint of the original inversion, and we can actually plot characteristics now. So 0, everybody starts at 0. And as you go through the phylogenetic series, if you move in the positive direction, that's a movement towards the telomere, negative direction towards the centromere. And here are the proximal breakpoints of inversions. And you can see a nice strong relationship there. Oh, and of the distal breakpoints. Again, nice strong relationship. And if you squint, you can kind of see they're kind of coming together, which suggests that they're getting smaller phylogenetic series as well, but not statistically significant. So this is the pattern, and there are lots of interesting nuances, and I'm happy to talk to you about them, but really, this is just the observation that leads into the cytogenetic mechanism, and that's what I'm going to focus on next. So, chromosome, 1 inversion, serial inverted. Going into meiosis 1, this is what you will look like. You have all of your homologous chromosomes and your sister chromatids. And if we classify them, remember we have the 2 inverted regions and 1 homosequential region shifted down the chromosome. And instead of forming a nice inversion loop that you guys see in textbooks, it forms inversion figure 8s. Now again, if you look at regions, the inverted regions for C and B. Now B is particularly interesting. If you know something about inversions, the reason that they say they suppress recombination is because of the formation of acentric and dicentric chromosomes. If we get a crossover event in region B, we form 4 monocentric chromatids. And these are they. Now, we've got 4 monocentric chromosomes, but we have a new problem. We have duplications and deletions. And if you want to do the accounting, you can see it's those C regions that have been duplicated in one case and deleted in the other. Now we're going to talk about fundamental facts of Drosophila meiosis. When you have asymmetric dyads moving into meiosis 2, it is a well-established fact that the shorter chromatid will always be, or will be preferentially recovered, not always, excuse me. So, we know this from overlapping inversions, heterochromatin displacement, compound chromosomes, ring chromosomes, well established. Now, so what we're going to ask is, what is the transmission of the serially inverted chromosome? And to understand that, we actually need to know some information about the behavior of the other chromosomes. So, first of all, in the respective dyads, these are the 2 shorter ones, they will always have an advantage over their partner. These 2 middle ones carry large duplications and deletions, the size of which for natural chromosomes are so large that they will cause lethal zygotes. So they have a disadvantage not at the transmission stage, but in terms of forming viable progeny. And so if we pose 2 parameters, one is the probability of forming an asymmetric dyad, and the other is recovery from an asymmetric dyad, we can actually write down the probabilities of the transmissions of the 2 non-recombinant chromosomes and the frequency of lethal zygotes from the cross. Okay, so this is the information now that we're going to introduce into a population genetic model. All right, so we're going to ask a simple question, classic question. What's the probability a mutant survives to the next generation? We're going to talk about an infinite population, and it's classically modeled as a Poisson process, and you can follow the number of generations it lasts, the number of copies it finds, so on and so forth. So I'm going to adapt it to a diploid because I'm particularly interested in the Mendelian, or segregation, we should say. So we say the probability of having k number of alleles in the next generation is 1, a function of the Poisson with a mean of 2 for the family size, and the transmission through the Mendelian mechanism, which in this case, if it was truly Mendelian, would be 0.5 per cube. We can write a recursion equation for that probability, and we have a nice probability, log probability of survival here. Now what I want to imagine you do is first think of a population that has a high frequency inversion in it, like any of the arms of melanogaster or chromosome 3 of pseudoobscura. And we're going to imagine a new mutant, and we're going to imagine the placement of new mutation is variable. Oh, I'm so sorry. First we need to modify this to incorporate lethal zygotic frequencies for C and the transmission Q. And we'll do that here. This is just those values from the previous 4-chromatin diagram that I showed you. Now imagine the population that is segregating inversion and 3 new mutants that come in. And they can be placed distally, medially, or proximally. And these are the consequent survival probabilities. Now it's important to notice that essentially everybody is going to go extinct fairly soon because of the lethal zygotes. However, the time that distal inversions are going to segregate the population is slightly more. Now we don't have to just limit ourselves to these 3. We can actually talk about it for all possible inversions from the 2 axes of size and position. And we can create a nice little surface. I know this is not lovely, but it's the survival probability of 10 generations, and we can actually kind of rotate a little bit. And you can see the first thing is that distal inversions tend to survive longer than proximal inversions. And smaller inversions tend to survive longer than larger inversions. Now, that's kind of a nice set of theory, and again, it can be applied to any population and for any set of inversions. This is just one scenario. But the really great thing is that we can actually test this. Now, we'll go to the historical record for pseudoobscura, for where we have 40 40 gene arrangements known for chromosome 3. They've also been observed through the years multiple times. Now first we're going to throw out all those common balancing selection inversions, about 6 of them. We won't care about those, we'll care just about the rare ones. And so of the remaining rare ones, we can divide them in half. Ones that we've seen once, and never again in subsequent samplings, and those that are seen at least twice, either 2 different ranges or twice at 2 different points in time. If we look at the jackknife distribution of the position and the size, position on the left, size on the right, for those rare single copy inversions, we have this distribution, which is a good kind of idea of what the mutational input is. And you can see the distal shift in those that have been seen twice, those that have persisted at least 2 samples. Same thing for size here, although it's less striking. Okay, so in summarizing, we'll talk about the 3 points that we wanted to lay out in the beginning. 1, in phylogenetic series, inversions tend to be moving distally. The number of inversions are at the end. When you have a cross and you generate asymmetric dyads, just from basic cytogenetics, always know that the chromosome carrying the distal inversion will always be overtransmitted relative to proximal inversions. Finally, if you incorporate those into a classic population genetic model, you can say that the new inversion spiral is a function of the relative size and position of the inversions in the population already segregating. And that fits very well with the empirical data. So, I'm happy to take any questions. I just want to acknowledge funding for this work is more than not true, and the key citations here. Not the full list of every inversion. Okay, thank you. No, I will say that my maps are not scalable combination, they're just in cytogenetic distances. The amount of size variation along the chromosome of the inversions might matter. I don't know how to answer your question other than to say, I started with a pattern that was observed in nature and showed that it was reproduced. I still don't know all the possibilities that could have been out there. If you look at other flies don't always have the distal clumping. And then, again, there could be a mechanism like you're suggesting operating in other species based on the combination map along the chromosome. In this case, I just don't know. Yes, ma'am.
Have you considered the gene conversion happening in their evolutionary?
I'm sorry?
What is gene flags? The gene conversion happening in their evolution region? Have you considered the slide activity of that?
No, I've not considered that. Other people have looked at gene conversion inside of inversion events, but not in the context of this mechanism. Was there another? All right. Okay. Great, thank you. Okay.